Yes, sir, it's visible.
Yes, sir.
Okay, I'll take my laser pointers like before. And okay, so shall we? Okay.
Yes, sir.
Yes, sir.
So if you remember last time we looked at how DNA gets replicated, and towards the end of the lecture we looked at some enzymes which act one way or the other on DNA as a substrate. Okay, so the first enzyme we looked at very quickly was ligase, and we said ligase is an enzyme which joins DNA together. Okay, but what it needs is a 3' OH and a 5' phosphate, and once it sees that, it will join it together to form a phosphodiester bond, and it can do what is called as nick sealing. That is, if two of the strands of DNA, one has a breakage, it will seal it, or sometimes if there are two separate strands but they are correctly aligned and they have a OH and a phosphate group, then it will seal that also. So it can also what is called as ligate two strands versus seal a gap in one of the strands. The next enzyme we looked at was what are called as nucleases, more specifically endonucleases, which we says can do what is called as clean cuts and give you pieces of DNA by cutting it in the middle, or sometimes just nick at different places if it does what is called incomplete cutting, and you can get DNA which has got little joints here and there, with gaps here and there. Okay. Then we looked at what is called as endonuclease, staphylococcal nuclease, which is very unusual that when it cuts, it leaves a phosphate on the 3' position rather than the 5' position. Then we looked at ribonuclease H, which is very unusual. It always detects a DNA RNA hybrid, and once it detects a DNA RNA hybrid, it cleaves off the RNA or it degrades the RNA, okay, and leaving only the DNA strand. Then we have what is called as nuclease S1 or S1 nuclease, which if it recognizes a single stranded DNA or single stranded RNA, then it will simply start breaking it up. It doesn't need to look at a hybrid. All it looks at is if there is something single stranded, it will break it. Okay. And then we looked at exonucleases, which cut either in the 3' to 5' or 5' to 3' direction. This example was 3', 5' exonuclease, okay, where it removes what is called as a nucleotide from the 3' end. Then we looked at what are called as phosphatases, and we said phosphatases remove phosphate groups which are present at the ends of DNA, either 5' end or 3' end, to give you what is called as phosphate-free DNA, and what is called as polynucleotide kinase, which if it sees a strand of DNA or piece of DNA with a 5' OH free, using ATP or any other NTP, will take a phosphate group from that and add it to the 5' end to give you a 5' phosphate. To close our discussion, yesterday we stopped before this. The last enzyme I want to talk about is what is called the template independent DNA polymerase. Okay, this is very unusual. Before that, if you remember, when we looked at DNA replication and we looked at DNA polymerase, we said it always needs a template, and it looks at the template and correctly adds the complementary base. Now this here is a very unusual DNA polymerase. It's template independent, but it does something very interesting. If it sees a strand of DNA, if it sees a strand of DNA, what it does is it goes to the 3' end or the 3' OH, and it simply adds nucleotides to that, irrespective of whether there is a template or not. Okay. Now the interesting part is, because it is adding at the 3' end, it is referred to as terminal deoxynucleotidal transferase. So it is only going to transfer to the 3' end. It will never add at the 5' end. Now the interesting part is, if I use say ATP here, deoxy ATP, then you'll only add A. So what I'll get is a DNA with a big tail which is a A. Okay, or if I put GTP, I'll get a big tail which is a G, and so on and so forth. Or if I put a mixture, it'll put it at random. Okay, so but what it does is it doesn't need a template, but it adds only at the end. Okay, and it'll add it randomly depending on which nucleotide is available for addition. Okay, so now with this little background, we are actually going to start what we started out to do, which is what is called as genetic engineering or DNA technology. Okay.
So,
Yeah.
sir,
Yeah.
I remember a process where poly A tail was added.
Correct.
So that template independent is only there.
Yeah, correct,
Then
correct. You must have seen in 11th or 12th when we talk
yes,
about eukaryotic RNA and how it is, modified. We say introns are removed, exons are kept, and then a poly A tail is added. Okay, that enzyme is also very similar to this. Okay, it just keeps on adding. Very specifically adds A. It doesn't add anything, but it adds a poly A tail. Okay, very similar to that. You are very right. Okay, so here we go. This is where our actual meat of the lecture starts. Okay, so we're going to use a word called cloning. Okay, and you must have heard this word of clone, this word here and there. Okay, so what does cloning actually mean? Okay, cloning means use of asexual reproduction to obtain organisms that are genetically identical to to one another and to the parent. Okay. Now this is in contrast to what is called a sexual reproduction, where you will always find that the offspring have got genetic diversity. Okay. They take some traits of the parents, but they are not identical to the parent, and neither are they identical to each other unless they are what are called as identical twins. Okay. Now, you have to also remember that when we use the word cloning, what we mean is the organisms that we get due to asexual reproductions are identical genetically. Okay, but because they are genetically identical, doesn't mean they'll also appear the same. Okay, sometimes what happens is even though you are genetically identical, the actual appearance and behavior is many times influenced by environmental factors, and therefore even though you might be genetically same, you might not look the same, but you are genetically identical. So if you are genetically identical, you are referred to as a clone. Okay, now we have seen this at many places. Sometimes in agriculture, we know that if you want, a plant which has got some features, and you want to retain that feature, what you simply do is cut the plant and plant it somewhere else, or you sometimes cut the plant and graft it onto another plant, and that way the genetics of that plant is maintained. You have also seen that in
So,
microbiology. Yeah, yeah.
so can
Yeah.
monozygotic twins be called as clones?
Yeah, identical twins. That is, twins which have started with the first fertilized egg, which actually splits into two and then goes on further, are said for all purposes to be almost clones. Okay, they are genetically identical. Okay,
But sir,
and that's why they are the
they
ones.
are not exactly genetically identical, right? Identical
Uh,
twins.
they
There might
are.
be
Okay.
like single base pair changes might take place, right?
Yeah,
Between.
that is, see, that change takes place a little later. But initially, when that splits, because they start from the same egg, they are technically speaking genetically identical. But you have to remember that even though they are genetically identical, they might not look at the same because of these environmental factors. There are a lot of changes which occur, which are called as epigenetic changes, or as you said, sometimes single nucleotide changes, which occur little later during the division of the organism, and because of that they don't end up being exactly the same, but you will find that they are very difficult to distinguish by genetic testing. When you do genetic testing of identical twins, invariably you can't pick them apart because they are so close to each other genetically. So technically you can always call them clones. Okay. Now, similarly, in microbiology, if you remember, when you do microbiology and you do what are called as cells growing on a plate and you get these little colonies. We know those colonies arise from a single bacterium, and you can take that little colony, put it into another nutrient broth and let it multiply, and you'll find all the bacteria there are identical to each other because they are derived from the same parent. And bacteria many times will reproduce asexually by simply splitting or budding, okay, and they will maintain that identity. But more closer to home, you also do photocopying or what you call a xeroxing. Okay, so when you are doing xeroxing, no, you are actually kind of cloning the original. Okay, so you take your notes, go to the fellow and tell him, मेरे को इसका xerox देना or photocopy देना, and you see that what you get, even though it is just slightly different paper, in terms of the material is almost the same. So such what is called as preservation of the identity to the parent is referred to as cloning. Okay. Now, this term cloning is also used in terms of genes. Okay, what this means in terms of genetic engineering is, if you take a foreign gene and put it into a bacterium, that means what? I take a piece of DNA which is normally not present in a bacterium and I put it into the bacterium, or I do that into any other cell. It could be a yeast cell or it could be a mammalian cell, and I do it in such a way that this foreign DNA is present now as part of the bacterium or the cell, and not only that, that when that cell replicates, this DNA which I have added also replicates, and you will get copies of that original DNA. Now what will happen is, if you allow these cells to replicate, which are carrying this foreign DNA or foreign gene, as they replicate, you get a large number of cells, and all of those will carry the same copy of the DNA that you have put into the original bacterium or the cell. When you do that, the terminology which is used is that you have cloned the gene. That means what? You have made lot of copies of the same gene identical to each other, but the way you have done it is by taking the gene and using a bacterium or a cell to do the replication, because the bacteria and the cells have all the enzymes which are needed for replication, just like we saw before, like DNA DNA polymerase and so on and so forth, which works without an error in most cases. Okay. The issue is, why would you want to do this? Okay, why would you want to take a gene or a piece of DNA which is present in one organism, put it into a bacterium or another cell and make a whole bunch of copies of it? Okay. Now there are several reasons we do it. One is, you might not know anything about that gene other than it's a piece that is there with you. So if you want to study it further and want to know the sequence of that little piece of DNA that you have or a gene that you have, one way to do it is make a lot of copies of it, and then once you have a lot of copies, take some sample and then send it to what is called a sequence determination, where it will be subject to some tests where the exact sequence of whether it is A, T, G, C, in what order will be determined. The other reason you might want to take a gene and make multiple copies of it is to kind of tag it, or what is called as label it. And once you have labeled it, you can follow it in terms of what happens to it in an organism as it is carried forward and expressed in terms of the message. Okay. Or you might actually want that gene for the express purpose of getting the protein out of it. Okay. For example, let us say this foreign gene is for human insulin, which you have put in E. coli, then what you want is for the E. coli to make human insulin. Okay, so you are interested in that protein product, and that's why you want the bacteria to grow, carry this gene with it, multiply it, and later on when it expresses it, you'll get a lot of human insulin, but this time made by a bacterium. More interestingly, sometimes what you might want to do is, you know a lot about this gene, and you feel that it is involved in some process, like let us say a disease, or it gives a unique property to an organism, and what you want to do is see whether that is true. And one way to prove that it is true is to do changes in the DNA and see what those changes reflect as in the organism. Okay, what is called as directed mutation. Okay, and that might be one reason why you might want a piece of DNA, so that you can later on change it very systematically and then study the effect of these changes as to what it does to the gene. Or you might want to do what is called as gene therapy. Okay, I produce a lot of one gene and then I use it to give it to a person who doesn't have that gene in order to cure that person of a genetic defect. Or I can do what is called as transgenics. Transgenics means where I take a gene from one organism and then permanently make it a part of another organism, such that now I have a new organism which has got some properties which normally it wouldn't have, but which it has got from another biological system. Okay, and there are several other reasons. I've given you some reasons why you might want to do this thing which is called as cloning, which is simply taking a piece of DNA, putting it into the organism and producing lots of it, or what you call as amplifying it. Okay. Now, what I've just told you with respect to this first slide and respect to second slide is shown here. Okay. This is how I started. Remember, I said in microbiology you do cloning. So you have a mixed bacterial culture, you put it on a plate, and if you dilute it enough, then what will happen is at each point on the plate there will be a single bacterium. Then you allow that to grow and you get these little colonies. But all the cells in these colonies are originally derived from one bacterium. So if you take each of these little colonies and put it separately in different nutrient broth, then you will actually have different clones of bacteria. Okay, all which are genetically different, but within this tube they are genetically identical. So here you are doing something same. What you are doing is, let us say I have got a mixture of DNA fragments. What I do is take that mixture and somehow put it into a bacterium, and then I allow that bacterium to grow, and I know that each of this colony which is present here is carrying a different piece of DNA. And if I were to take that very colony and multiply it, I'll get a whole bunch of those cells, but each of them will carry the same piece of DNA which I managed to put when I did that first step. So this is also referred to as what is called as cloning, but this is cloning of DNA, but this is cloning of the bacteria itself. Okay. Now, how do we do this? Okay. And what we and the process by which we do this is what is called as a cut and paste technology, or what we call as recombinant DNA technology. So in essence, what you do is take the piece of DNA which you want to amplify, and that is referred to as DNA to be cloned. Sometimes you will find a book will say that is target DNA. Sometimes the book will say foreign DNA or foreign gene, whatever terminology you want to use. But the idea is, this is a piece of DNA I want to amplify. So what I do is, if I try to put this DNA directly into a bacterium, or directly into a yeast cell, or directly into an animal cell, I many times find that this piece of DNA will simply be degraded when it gets into the bacterium or get into a cell. So that way of putting a DNA into a cell and making it amplify usually doesn't work. What you tend to do is use what is called as a vector. A vector is nothing but a carrier which carries this piece of DNA very safely along with it. Okay. So what you do is, you take a carrier and into that carrier you introduce this piece of DNA to give you what is called as a recombinant carrier or recombinant plasmid. Okay. I use the word plasmid here because here the vector was a plasmid, but it could be any other vector. Okay. What is important is, I have an original vector. If I put this piece of DNA into this vector, I will have a recombinant vector, a vector which is now carrying another piece of DNA. Now what I do is, this vector has a property of going into a cell and safely being there without getting degraded, and when the cell multiplies, it will also get multiplied. So what you do is, you take this recombinant plasmid and put it into a bacterium, for example, by the process which is called as transformation. So you transform this bacterial cell from what it was to one which contains this plasmid. Now once you do that, you replicate this bacterium. Now as it grows, this piece of plasmid will also get replicated, and then you'll have a whole bunch of bacteria all now carrying this plasmid, but this plasmid has that piece of DNA that you have, and then later on all you have to do is break open this DNA, and when you break open this DNA, you'll get back this little recombinant plasmid, but now instead of one recombinant plasmid, you'll have millions of them. And what you simply do is do the reverse of this process that you did here, cut it back to give you back this DNA, but this time you'll get millions of copies of DNA. Okay? So, because of the way this is done, this is called as cut and paste technology. Okay? And the reason it is called cut and paste technology is you need to cut something to get your DNA. You need to cut the vector so that this DNA can be put here. Then once you do that pasting, you seal this to get the recombinant plasmid, and then you simply put it into the bacteria and multiply it. Okay? So easier said than done. Okay? Now we are going to see how this actually is done in a very systematic fashion. Okay? So we are going to see how we do this isolation of DNA, how we do the cutting of the vector, then we'll see how they are put together and ligated or what is called as joined. Then we'll see how they are introduced into a bacterium, and then how you pick the bacterium which contains this fellow and then multiply it, and then later on isolate the DNA. So to understand that, the first thing we are going to start to look at is a group of enzymes which I kind of talked about but I didn't talk about. Okay, these are referred to as restriction endonucleases. Okay, now in the previous few slides we talked about an enzyme which was called an endonuclease. Remember? And this endonucleases are enzymes which cut DNA somewhere in the middle of the DNA. Okay, and remember, they can make pieces of DNA or they can make little nicks in the DNA. Now, restriction endonucleases are a subtype of endonucleases which have got very specific properties which allow them to be used very intelligently to do what we want to do. Okay, but what are these restriction endonucleases? Okay, they are from bacteria. Okay, they are isolated from bacteria, and in the bacteria they do something very interesting. They cleave any foreign DNA which comes in. Okay, and where does foreign DNA come into a bacteria? For example, if this is a bacteria and this is its own DNA, this bacteria can sometimes be infected just like we get infected by viruses, and viruses which infect bacteria are referred to as bacteriophages, if you remember. So when these bacteriophages infect the bacteria, they'll enter the bacteria and they release their DNA, and this DNA will then be made into proteins and all the other things by the machinery of the bacteria. Now bacteria doesn't like this foreign DNA and doesn't like getting infected. So what it has is some defense system, and this defense system are these restriction endonucleases, which as soon as they see this foreign DNA, they cut it. Okay, and they degrade it so that the bacteria doesn't get infected by the virus. Okay, now the interesting part is this restriction enzymes do not cut this DNA in a random fashion, and they cut it very specifically at certain places when they see a certain sequence. Now it is entirely possible that that sequence might also be present in the bacteria's own DNA. So what the bacteria does is, so that these restriction enzymes don't cut its own DNA, it modifies the same sequence which is present in its DNA by a process called as methylation. Okay. So just to give you an example, let us say this restriction enzyme recognizes a sequence which is ATTA. Now wherever ATTA is present in bacteria's own DNA, you will find one of those bases, A, T, T or A, will be methylated, and because it is methylated, this restriction enzyme will not be able to recognize, and therefore the bacteria's own DNA is preserved and is not degraded by the restriction enzyme which it is producing. Okay, so going back, so these restriction enzymes, which are part of what is called as restriction methylation system. Okay, why did I use the word methylation? Because whichever organism produces a restriction enzyme, it also makes sure that its own DNA never gets degraded by that enzyme by methylating it. Okay. Now, lot of organisms are known to produce restriction enzymes, and the name of a given restriction enzyme is usually indicative of where it is obtained from. For example, there is a restriction enzyme which is called as EcoR1. Okay, in EcoR1, E and Co stands for Escherichia coli. R stands for the strain of E. coli which produces it, and this 1 is the enzyme which was discovered in chronological sequence. Okay, so for example, if another enzyme was discovered in the same strain, it would be called EcoR2, and so on and so forth. Similarly, BAMH1 is obtained from Bacillus amyloliquefaciens, strain H, and this is the first enzyme which was found in this bacteria. As I said, it protects the bacteria from bacteriophage infection, and the bacteria protects its own DNA by methylating those sequences which are recognized by this restriction enzyme. Okay. Now, something very interesting is that as of today, if you were to look more and they recognize distinct sequences, and the length of the sequences are anywhere from 4 to 8 bases in length. Okay, so what these restriction enzymes will do is, if you give them a piece of double-stranded DNA, they look at it, and only when they see a certain sequence which is of length about 4 mer or length up to 8 mer, they will recognize it and they will cut it. Okay. But what is unique about the restriction enzymes that we are talking about is that they always recognize a sequence which is palindromic. Okay. And depending on which sequence you want to cut, you can buy that restriction enzyme from a company. Okay, so companies sell all these restriction enzymes, and they'll tell you which restriction enzyme cuts which type of palindromic sequence. And depending on where you want to cut a DNA and how you want to cut a DNA, you can buy. Okay. Now the issue is, what is a palindrome, or what is a palindromic sequence? A palindrome in English or regular English is any word or a group of words or a sentence which reads the same in either direction. Okay. For example, here I have got a list of palindromes in increasing complexity, starting with, say, a single alphabet, which either which way it will be the same. Then let us say Eve. So if you read it this way is also Eve, or say radar, or reviver, or rotator. Here is a a little statement: Madam, I am Adam. Okay, and if you read the other way, it also reads, Madam, I am Adam. Then you have stepped not on this path. So another famous palindrome is Abel was I, ere I saw Elba, or a man, a plan, a canal, Panama. Okay, or a very big one, on a clover, if alive, erupts a vast pure evil, a fire volcano. Okay, now there are a lot of competitions also. People have actually written palindromic poetry, palindromic stories, where if you start reading either which way, it is made up of the same alphabets appearing in the same sequence. Okay, but when it comes to DNA, the meaning of palindrome is slightly different. Okay, so I'm going to show a section of DNA with some sequences which are very interesting. Okay, so look at this top pieces of double-stranded DNA. Okay, if you look at this top piece of double-stranded DNA, you find that the sequence here T, T, A, G, C, A, C is repeated again. Okay, so here also you find T, T, A, G, C, A, C. When something like this appears on a DNA, you call these direct repeats. Okay, that means it's the same sequence repeated on the same strand of DNA in the same direction. Okay, now if you presume this is 5' end and this is 3' end, so as I go from 5' to 3', I see TTAGCAC, then following it, I see another TTAGCAC. So such sequences which appear at tandem are called as tandem repeats. If they appear after little gaps, they are called as separated repeats, but they are direct repeats. Okay. As opposed to that, here is another one which is called as a mirror repeat. Okay, now look at this sequence here. Again, I am reading from the 5' to the 3' direction. Now, when I do that, see what happens here. I go T, T, A, G, C, A, C. And then let us say if I were to put a mirror here, I find the same sequence repeated again. Okay, but in the reverse direction. That is C, A, C, G, A, T, T. It looks as if I put a mirror here and I am simply reflecting these bases on this side. If such a sequence is seen in DNA, it is referred to as a mirror repeat. Okay. So what's the difference between a direct repeat and a mirror repeat? Direct repeat is same sequence appearing in same order, one after the other or with a gap. A mirror repeat is the same sequence appearing as if it is mirrored but on the same strand. But relative to these two, look at this piece here. Now look what has happened. I have got a sequence which is TT, AG, CAC. But now look at what happens on the other strand. Now look, on this strand I am moving from 5' to 3' direction here. So in the 5' to 3' direction, I get T, T, A, G, C, A, C. Then I go further and it is G, T, G, C, T, A, A. Now I come to the other strand and I again go from the 5' to 3' direction. I find that the same sequence is actually repeated here, but it is in an inverted fashion on the other strand. Okay. So look, T, T, A, G, C, A, C, just like you did here. And then continuing G, T, G, C, T, A, A, just like you saw here. Okay. So such sequences are referred to as palindromic sequences as far as DNA is concerned. Okay. Now, in normal English, this would be called as a palindrome. Okay. Remember, you saw the previous slide. You look at a word and it reads the same as if you put a mirror in between. Okay. So, if you look at the DNA, a mirror repeat should have been called a palindrome, but that is not what they do. The reason they say this is not a classic palindrome is because when I read DNA in this direction, it is 5' to 3, but when I look at this piece and I look at this direction, it is actually 3' to prime. So I am not technically doing things correctly. But here, look, I am reading from 5' to 3 and I am here also I am reading from 5' to 3. So this is actually true palindrome according to DNA. So in DNA, what are called as inverted repeats are referred to as palindrome, and it is these type of sequences which are recognized by restriction enzymes. Okay, and when they see this sequence like this, they cut the DNA. Okay, and very beautifully they cut the DNA usually on both strands, because either which way the DNA goes into the enzyme, it looks the same. Okay, so you can imagine if there's a long piece of DNA and this is a sequence in between here, whether I look at it this way or I turn it around, it look way and it look the same. So as far as a restriction enzyme is concerned, it will be able to cut either on this strand or on this strand, or on, actually it does it on both strands. Okay. Now in this world, there are different types of restriction enzymes which are available, and the one which we are interested in is referred to as a type 2 restriction enzyme. Okay. There are 4 types of restriction enzymes, all which have this property of recognizing a sequence and cutting the DNA. But they differ in terms of how they recognize a sequence and actually where they cut. But what is interesting about a type 2 restriction enzyme is that they recognize a palindrome, which I just told you, and they cut within the palindrome. Okay, and that is what is important. Okay, when I know an enzyme recognizes a given palindrome and I know it cuts within the palindrome, I know for sure how it is going to break the DNA. As opposed to, let us say, a type 3 enzyme, which say it recognizes a asymmetric palindrome, which means what? A palindrome which is present with gaps in between, and it cuts somewhere 25 to 27 bases from that site on the 3 prime. So now I'm not sure where exactly it's going to cut the DNA, so I will not know what I'm going to get. Now the importance of the restriction enzyme is because it recognizes a specific sequence and it cuts within the sequence. I know that when I take a DNA which contains a palindromic sequence, I know exactly how it is going to be cut by a given restriction enzyme. Okay, to give an example, here is a restriction site. Okay, now remember this is a 6-mer restriction site. Okay, 6-mer means it has got 6 bases, which is part of the palindromic sequence, and this sequence is C GAATTC on this strand, and when you go to the other strand and read it in the same direction, it is again GAATTC. Okay, so this sequence is now being present as part of a big piece of DNA, and if this sequence is recognized by an enzyme, it is going to be cut by that restriction enzyme. Okay, now here are examples of different types of palindromic sequences, but what I have shown here is a sequence on one of the strands, and if you look very carefully, you'll realize that the other strand also has the same sequence. Okay, so going back, remember the sequence. See, GAATTC. On the other strand also it will be GAATTC. Okay. Now this is the one which is recognized by the enzyme called EcoR1. And if you look at this sequence here, I put a little slash. What that slash tells me is that this enzyme EcoR1 will recognize this sequence and it will cut between the G and A on both the strands. Okay, so here is G and A, so the EcoR1 will cut here, but it will also cut here, and when it cuts here, I will get 2 pieces of DNA. Okay. Similarly, here are other enzymes and they are also cutting. Okay, so some cut on this end of the palindromic sequence, some cut on the right hand side or the right hand end, and some cut right in the middle. Okay, depending on how they cut, you get what are called as sticky ends or what are called as blunt ends. Okay, I'm going to show that in the next slide. Okay, now if you look very carefully here, here are the names of the restriction enzymes, here are the organisms from which they are derived, here is the length of sequence that they recognize, and depending on how they cut, whether they generate what are called as sticky or blunt ends. I'll tell you what sticky and blunt end is in a second. Okay, I'll give you 3 examples of 3 restriction enzymes. So EcoR1. Okay, so EcoR1, if you remember, recognizes GAATTC and cuts between GA. So when it cuts here, I get 2 pieces of DNA, but look at how the pieces are. The ends are actually projecting here on this side, here on this side. So here you have what is called a 5' projection. Here you also have a 5' projection. But the beautiful thing is, this sequence here is complementary to this sequence. So if I were to bring this piece close together, there's a tendency for this to base pair with this. That's why they are referred to as what are called as sticky ends. Okay. Look at this restriction enzyme. It recognizes a sequence CTGCAG, which is also palindromic. CTGCAG. But see how it cuts. It cuts between GA here and GA here. So when it cuts, see what you get? You end up getting again sticky ends, but the projection is on the 3' side rather than on the 5' side. But again, if you look at these 2 sequences, if I bring this piece close together, again there will be base pairing. Okay, and that's why they are also referred to as sticky ends, but this is called as 3' overhanging, and this is called as 5' overhanging, or 5' projection or 3' projecting. As opposed to these, yeah.
So
Hello.
sticky
Yeah.
ends means they can join again, or it's something else.
Yeah. See, technically, because of this nature, no, it's almost like, you know, a Velcro thing. Okay, here I've got these fibers, and here there is Velcro open. So if I bring this together, because of this complementarity, you know, they can actually even come back and sit here. The thing is, the enzyme has cut it here. Okay, but if I bring it together, the DNA will not join and become intact, but it will appear to be intact, but there will be little gaps here. Okay, because the restriction enzyme has cut it, and that tendency for it to come back and stay kind of, you know, interacting with each other results in this name, which is called as sticky end. Okay, is that clear?
Yes, sir. Yes, sir.
Now, see, what is very interesting is this. Let us say I had 10 molecules of DNA of this type, okay? And I cut it with this. I would get 10 of these type of pieces, and I'll get 10 of these type of pieces, correct? And then what I will find is, because of this tendency for stickiness, at randomly, piece of the first, let us say, A piece can interact with this of the D piece. F of this can interact with D of this. Okay. The reason it can do so is because the ends are sticky. Okay. Are you seeing what I'm trying to tell you? So if I have a population of these pieces and I get a population of these, when they are sitting in the test tube, they will have a tendency to come together, but they will come together at random because of these being identical and the tendency for them to base pair. So I can actually get hybrids, okay? But all the pieces will actually have the same sequence because I'm starting with this, but you have a tendency for stickiness, okay? Now relative to that, look at this restriction enzyme. It cuts right in the middle. When it cuts right in the middle, what I end up getting is two pieces, but now I got flush ends or blunt ends. Now, even if I put them together, they have no tendency to stick to each other, okay, because there is no overlap, okay. So depending on which restriction enzyme I have, I can get what are called as sticky ends, and they are referred to as 5' sticky ends or 3' sticky ends or 5' projections or 3' projections or 5' overhangs or 3' overhangs, but they all have a property where the two pieces still have a tendency to come together, although they will not join together, but they will tend to interact with each other. And that tendency is not there in a restriction enzyme, which does what is called as a blunt end cutting or what is called as a flush end cutting. Okay. So this is something like, you know, if you remember when you were little, you used to get jigsaw puzzles, little puzzles which have got different cut shapes, and there are two ways to join together those puzzles. One, you look at the picture and try to join them together, or what you end up doing is making sure the complementary pieces, no, where there is a gap in one but there is a projection in the other, you put it together. Okay, so these are of that type. Now imagine you have a puzzle where all the pieces are exactly the same shape, but there is no projection. That is, all the pieces are little squares, and you have no picture to guide you. It's very difficult to put together that puzzle. Okay, are you seeing? Okay, so a blunt end is always more difficult to handle because these two won't come together, but these two will come together. Okay. Now, within the restriction enzymes also, there is something very interesting that sometimes happens. Okay. Uh, there are two restriction enzymes. They tend to recognize the same sequence. Okay. One is called as AC651 and the second is KPN1. But the difference between the two is, even though they recognize the same sequence, they cut at different places. Okay. So when I have two enzymes which are obtained from different sources, which recognizes the same sequence but cut at different places, they are referred to as isoschizomers. Okay, isoschizomers. But every once in a while, you also find that two different enzymes from two different sources recognize the same sequence and also cut exactly in the same fashion. If you have two such enzymes, fortunately, they are referred to as perfect isostereomer. So what is the difference between a regular isostereomer and a perfect isostereomer? A isostereomer recognizes the same sequence but cut differently within the sequence. And a perfect isostereomer not only recognizes the same sequence but also cuts exactly the same way. Okay. Now, going a little further. Okay. Now, presume for a second that you have a long piece of DNA. Okay. And in this long piece of DNA, somewhere in the middle of this piece of DNA is the gene that you are interested in. Okay. So you see these three things here. I've just shown three, but it could be little long and whatever length you want. But what is important is this is a piece of DNA that you are interested in cloning. Okay, remember? Go back to that. I'm going to go back to that little slide we started with, where you said, okay, I want to clone stuff. Here is that little piece of DNA I want to clone. Okay, so I want to cut it out. Okay, so see how I do that. Okay, so I'm going back through this restriction enzymes and I'm coming back to this side, and that little piece is here. Now what I find is that flanking that piece of DNA are two palindromic sequences. Okay, and see the sequence CTTAAG. Again, this is CTTAAG. Here also it is CTTAAG, and again CTTAAG. Okay, I realize that this sequence is recognized by EcoR1. So if I take this piece of DNA and I treat it with EcoR1, because EcoR1 cuts between the A and G, okay, at both places here, I'll find that if I take this piece of DNA and treat it with EcoR1, I'll get three pieces. One will be this piece, which I'm actually not interested in, which is derived from this part. So it is kind of this part here. See this? Because the cut occurred here and cut occurred here. The other piece is this one, which also I'm not interested in, which occurred because of the cut which is occurring here and the cut which is occurring here. But look at this third piece. The third piece has got my gene of interest, but which is flanked by two ends which are sticky. Now, if I take and throw these two things away, now what I've done essentially is from a long piece of DNA, cut out the DNA that I'm interested in. Okay, so now I've got the DNA of interest cut out from a source DNA, and that I'm going to try and now put it and join it into a vector. Okay, so we are going to leave restriction enzymes at this point, knowing what they do and how they can be used to very specifically cut out a piece of DNA. The only requirement for this to work is that if you have a gene of interest, what you require is it should be flanked by two sites which are cut by the same restriction enzymes. And if you are lucky enough, in many cases you tend to be lucky, and if you are not, there are ways to get lucky. But at the basic level, if that is true, then you just take that restriction enzyme, treat your piece of DNA, and you'll get a whole bunch of pieces which look like this, which are all pieces which are carrying your gene of interest, but which have what are called as sticky ends. Okay, so now let us look at this thing called the carrier, or what are called as plasmids. Okay, there are different types of vectors, but I'm going to start with the simplest of vectors, which are referred to as plasmid vectors. And what are plasmids? Plasmids are basically pieces of DNA which are present in a bacterium other than its own genome. Okay, so if you remember, we at one point when we were talking about replication, we said E. coli contains DNA which is around 4 million bases long. Okay, this is that piece of DNA. Okay, assuming this is E. coli, this is what is called its genomic DNA. Now, many times, many bacteria, in addition to having their own DNA, they also carry extra pieces of what are called as small circular DNA. And this DNA replicates independently of this piece of DNA. And such pieces of DNA, which are actually not part of their regular genetic makeup, but which are all present in the cell, are referred to as plasmids. Okay. Now we also have something like that. Okay, you might not realize that, but you know that in every cell of ours, okay, except a few types, we have DNA in the nucleus, but many of our cells also have mitochondria which carry their own DNA. Okay, so that mitochondrial DNA in human cells is referred to as non-chromosomal DNA or extra chromosomal DNA. Bacteria also do that. They also carry this little pieces of DNA. But why do they carry this piece of DNA? The reason many times bacteria carry this piece of DNA is because this DNA has got some properties which allow the bacteria to have some advantage over the competitors in the environment in which it is growing. Okay. For example, some plasmids are called as F plasmids. What they do is, if that is present in a bacterium, they allow the bacterium to go through a process of what is called as conjugation. That means what? Now the bacteria can actually exchange DNA with other DNA and do what is typically referred to as a type of sexual reproduction. Or I can have a plasmid which is called as a R plasmid or resistance plasmid. What do these plasmids do? They contain little genes which, when they are expressed through transcription and translation, make proteins which give the bacteria resistance against antibiotics or poisons. Okay. For example, one such plasmid codes for an enzyme called beta-lactamase. Okay, and what does beta-lactamase do? It breaks a beta-lactam ring, and beta-lactam rings are present in many of your antibiotics like penicillins, cephalosporins. So if I have a bacterium which carries a plasmid which is able to produce beta-lactamase, then it will not be sensitive to all of these antibiotics which contain a beta-lactam ring, and that gives it an advantage over other bacteria which do not contain a plasmid. Okay. Another bacteria might have what are called as coal plasmids. They tend to contain genes which code for proteins which are bacteriocins. These are proteins which kill other bacteria by making many times pores in that other bacteria, and therefore I kill off my competition. Okay. Then there are plasmids which tend to contain enzymes on them, and these enzymes allow that plasmid to many times degrade compounds which are present in the environment, and not only degrade, use them as a food source. For example, there are bacteria which are known to produce enzymes which are able to metabolize, say, toluene or salicylic acid or even benzene or camphor, which is present in the environment, and then use them as a food source, okay, which gives them an advantage over other bacteria. There are other bacteria which contains plasmids which allow them to become virulent. Okay, for example, there is a bacterium which contains what is called as a tumor-inducing plasmid, and this allows that bacterium to infect plants and cause what is called as crown gall disease. Okay, and the bacterium which contains that is called as Agrobacterium tumefaciens. Okay, so the presence of this little plasmids here can provide an advantage to a bacterium, and therefore it carries it. For example, as I said, it allows it to conjugate, or it gives resistance, or it produces colicin, or it produces enzymes which allow it to metabolize different compounds, or it allows it to be virulent. So whatever be the case, these plasmids have a utility for the bacteria. Now, these plasmids are one of those agents which can potentially be used as a vector to carry your DNA of interest. Okay. Now see, normally a plasmid comes into the bacterium carrying a gene of interest to the bacterium. For example, a gene which gives resistance or gene which produces enzymes. gene which produces virulence factor. What I can potentially do is take a plasmid and manipulate it, somehow cut it out and put my DNA of interest and put it back into the into the bacteria. If I can do that, I can use this plasmid as a carrier or what is called as a vector. Okay. Now that is what people do. Okay. But what is required for a plasmid or a vector to be a good vector is that it needs to have some properties. One, it should be able to replicate on its own inside the host cell. Okay, just like a plasmid can. The other thing is very interesting. It must possess a unique restriction site. Okay. Now, what do I mean by this? We just look at restriction enzymes, right? And we realize that they recognize palindromic sequences and they cut very specifically. So, for a vector to be a carrier of DNA, it should also possess one or more palindromic sites which are present preferably only once in that entire plasmid or in entire vector, so that I can cut very specifically at one point without making bits and pieces of that vector. Okay. Now, what should also happen is that this vector, when I add a piece of DNA to it, that addition of DNA or what is called as making the plasmid now a recombinant plasmid should not interfere with its ability to replicate inside the host cell. That means that even though I do some modification to the plasmid by adding a piece of DNA, it should still be able to replicate. The other thing is it should possess some gene which I can use later to what is called as identify those plasmids which are different from the original. Yeah. Hello. Yeah.
Sir, sir, we have a microbial genetics lecture at 2:15.
Okay. So what I'll do is I'll stop here at this point and we'll continue next time here. Okay. So I'm going to stop my sharing, and if you have any questions, you can ask now. Okay. Any questions at this point before I close the lecture? Okay. So we'll go further, and maybe as we go along and we manipulate the DNA a little more, more questions will arise. Okay. So this till this point, all what is important is we have learned what are restriction enzymes. We know they cut very specifically within a palindromic sequence, and they can give you what is called as sticky ends or blunt ends, and you can use that feature to cut out a piece of DNA. And we have just started to look at how we are going to carry that DNA into the bacterial cell by using what are called as plasmids, which have the ability to replicate inside a given bacterium. Okay. So I'm going to stop here for the day. Okay. Is that
Yes,
okay?
sir.
Okay. So I'm stopping my
Thank you,
recording.
sir.
Yeah.
Thank you, sir.
And